Thank you very much. I'm from the Moffitt Cancer Center, which is here in the state of Florida, about 4 hours across over on the West Coast. So welcome to Florida. And if you notice, it's 85 degrees out right now, so we're about halfway through summer right now. So just to let you know. No financial disclosures, funding from the state of Florida, as well as through a variety of grants to the NCI. So our group has been really largely focused on a plethora of areas within in radiomics of lung cancer. We know it's a major problem. We know it's high incidence, high mortality, poor survival. And I just published a really large piece in CEBP this month with my colleague from Carmanos on progress and priorities related to lung cancer. And we spent some time highlighting really a variety of things that we need to do to improve this lethal disease that has a substantial public health burden. And really the three things that are directly related to what we can do here in ECOG-ACRIN, what we can do with radiomics, is everything from precision risk assessment to improve diagnostics. And what's really particular salient in early detection is reducing false positives and thinking about overdiagnosis. And of course, we've talked a little bit today about predictive biomarkers of both negative and positive responses. And I'm actually going to skip through some stuff so I can get to, I added some slides that are not related to the NLST about really important things we could do, some algorithms, pre-treatment algorithms, using quantitative imaging and radiomics for negative and positive responses. So when you talk about the cancer control continuum, our group conducts radiomics across everything except for prevention. And when we're thinking about early detection setting, we're largely focused on primary and secondary. So why screen for lung cancer? I think everybody knows. We know the majority of lung cancers are diagnosed at late stage, and survival is very poor. 4.3 overall, it's about 19% survival overall for lung cancer, but when you look at late stage, it's a 4.3% survival. So the goal is really to move the diagnosis from the red to the green, identify more early stage patients where we can treat them surgically and have better chance of cure. There's been 3 major lung cancer screening trials that showed the efficacy of using low-dose CT, the NLST, which I think everyone's familiar with, which was conducted here in the United States. And 2 other trials came out this year, the MILE trial and LUCY trial, all showing the benefit of screening with LDD-CT to reduce lung cancer mortality. And we know that the Nelson trial, which was presented last year in Barcelona, no, it would have been last year, not Barcelona was this year, I think it was Toronto, that showed that it is, Nelson trial, which is a larger European trial, showed that LDCT is effective in reducing lung cancer mortality. But it comes with a cost. We know that LDCT is going to detect a lot of nodules. In fact, in the NLST, 24% of individuals screened with LDC had a positive finding, meaning a 4 millimeter nodule or greater. Vast majority of those were false positives. Only 4% were cancer. And of course, as similar to what we see early detection of breast cancer and prostate cancer, you have to worry about overdiagnosis. And a variety of estimates, anywhere from 10 to 65% of lung cancers in the early detection setting are believed to be LDCT or overdiagnosed. So the 3 areas that our group has been focused on, and I'm not going to try to make the claim that we're the only one. A lot of people are working this. We were very early adopters of this. We really were one of the first groups to get into the NLST images and start extracting quantitative imaging and doing these analyses. So a lot of the data I'm going to show is really just sort of the breadth of work that we focused on at the NLST. So one is risk. If a patient comes in today with a nodule, a 5 millimeter nodule, guidelines tell you leave it alone, come back in 12 months. So can we look at that nodule today and determine whether it's going to evolve and be diagnosed as a cancer in the future? A diagnostic. If a patient comes in today, can we use a non-invasive quantitative imaging algorithm to predict if this is cancer right now? And again, overdiagnosis. Now that you do have a diagnosis, can we understand the behavior? Patients that have a very aggressive phenotype that would require additional follow-up, adjuvant therapy, any other modalities to hopefully reduce risk of recurrence, versus something that is very indolent, less aggressive, where you may just have to wait and see and put them into a surveillance clinic. So this is our toolbox. I don't think I had to dig too much into this. We saw a lot of how this is done, where we can take standard care images and quantify them using either quantitative features, radiomic features, or semantic features. These are actually what the radiologist will call and uses a lexicon of these things. And these numbers are sort of arbitrary, especially when you talk about conventional features, because there's really no known what the highest level is. They can be into the tens of thousands, because you could take an image, you could warp it, you could invert it, you could filter it, you could invert it, you could do a variety of things to it to calculate a whole array of different features. And then deep learning, where you can get into thousands, tens of thousands, hundreds of thousands of features. So risk prediction. So our first work started on this really many, many years ago, right after sort of the term radiomics was coined. And that term has actually only been around for about 8-9 years. And we published this in JTO, where we looked at baseline screens in the NLST. We did a nested case control study, extracted baseline radiomic features, and used that to predict cancer among those that had a positive screen, a 4 millimeter or greater nodule. And this is what we used. We used a random forest classifier, found a most parsimonious model set of 24 features with an AURC, here we go, 0.83. We showed that this is superior over using McWilliams or a volumetric measurement. Building on this work, there's been a tremendous amount of interest in understanding dynamics. As nodules change in time, how does that inform the model? So what we did is include radiomics. So we went back to the original cohort and looked at delta features and changes in features over time. But we also looked at size classes, because we know when you look at the sizes of the nodules, those patients that eventually get diagnosed as cancer versus those that are what are called the nodule positive, the distribution of their nodules are much different. So we did two analyses. We looked at size classes and looked at changes in deltas. So there's the cohort, the red boxes meaning the delta or changes in it. And we see generally when you look at any given size class, the black line representing using the combination of both delta features and baseline features, we see better improvement in our models. It's for any given size class. And so when you compare that to sort of where we started with that JTO paper, we found a model of just 5 features with an AURC of 0.88. So it's showing just head-to-head by changing our algorithms, by improving our pipelines, our analytics, we're actually showing that we're seeing these modest improvements. And this is purely radiomics. This is not even considering any sort of clinical features into these models. So a lot of the work that I just showed was all really quantitative features. We also wanted to explore these semantic features. Semantic features have existed as long as cross-sectional imaging has existed. These things sit in radiology notes. Radiologists have used these things, but in a non-standardized, non-uniform way. So what we did is we developed a lexicon, what was already existing out in the literature, and developed a new sets of these semantic features and applied them to the NLST. And we published this paper a couple years ago in Radiology. Now, on this, we focus on the negative scans. Why the negative scans? Well, for 2 reasons. One, these are patients that have 4 millimeter or less nodules. We're talking about 1 or 2 pixels or voxels, very small nodules. So they're really sort of difficult to quantify using conventional radiomics. Now, deep learning could probably do a good job with these, but conventional radiomics, where you're looking at a region of interest and extracting information, it doesn't work as well. So we used this lexicon. The other reason why we looked at those is that we identified that patients that have a negative baseline scan today and are diagnosed sometime in the future, a year or 2 later, actually have a more aggressive cancer compared to those patients that have a positive nodule today versus those in the future. It's sort of similar to what is seen in breast cancer. And so this was an observational analysis that we published in PLOS One. So when we focus on those baseline nodules that were 4 millimeters or less, and looked at the risk of cancer in the future, we saw that we're finding really large AURCs of 0.94 with substantial sensitivity and specificity. And these were things that sort of make sense. And we did some model building approaches to sort of narrow this down to what was the most informative. And there are things like emphysema attached to vessel. We know upper lobe is where a majority of lung nodules that end up being cancerous are diagnosed. Porter definition and concavity. These things were all sort of this existed in the radiology note for years. We all knew about this from the radiology world, but we took this and applied it in a study design and showed proof of principle how powerful these things are. I'll skip over that. The next is diagnostic. So patient comes in today, can we use a non-invasive approach to diagnose them or give a probability of diagnosis? And so we used the same cases and controls, but we obviously looked at the time they were diagnosed. So we didn't look at the baseline screen, we looked at the screen where they were diagnosed. And we also did some clinical benefit analysis. We did a variety of analyses. I'm not going to go through the whole method, but when we look at all size ranges, you basically focus on the red line on the left and right. Red line is using both radiomics and clinical features combined. That we're getting a pretty decent classifier of 0.82. So we have an AURC of 0.82 as a diagnostic descriptor, and you see that it has a better clinical benefit than just using clinical factors. And this was true for whether we use all size ranges or if we looked at 4 to 12, 12 to 30. So overdiagnosis, where I'm at with time. Okay, I'm going to skip ahead because we're doing a good job with overdiagnosis, just trust me. I want to get into something that I think that is particularly salient to ECOG-ACRIN, and this is not related to NLST. Predictors of immunotherapy response, and we've talked an enormous amount of time today about immunotherapy response. And we've done some preliminary analysis. We published a paper earlier this year showing that we can predict patients that are going to develop hyperprogressive disease. Hyperprogressive disease is a doubling in tumor size in less than 2 months, and it's a very lethal phenotype. And it's about 8% of patients will develop hyperprogression. And this is immunotherapy-induced hyperprogression. Because these patients we show have stable nodules, stable cancers before we're treating them with immunotherapy. After the onset of treatment, that these things double in size, at least double in volume. And it's not a pseudo response because these patients continued on therapy and the tumor continued to grow. So these were true responses. But what's important about hyperprogression disease is that these patients die within 12 months. It's a very, very lethal outcome. Luckily, it's very rare. And so biomarkers identifying them are a great value. Building on that work, we moved forward into treatment response across the plethora of what we would expect for phenotypes of outcomes for immunotherapy. And I think we've already heard about this today, why we need biomarkers to predict response. We know that if you take a given biopsy anywhere on the lung, that you can find different levels of PD-L1 expression. And PD-L1 is even though it is clinically approved, it is not the perfect biomarker to predict response, because we know that patients across ranges of PD-L1 will still respond. So what we did is we built this large consortia of patients from Moffitt, from the VA, divided up into a training test and validation cohort, did a variety of analysis, did a decision tree, did an ensemble approach, and identified four risk groups by using basically in a decision tree using one radiomic feature and using 2 clinical factors, and you could see those in there. And then when you take that and you plot that into Kaplan-Meiers, we see the 4 risk groups sort of pan out. In the training group, you see that gold group at the bottom. These are all patients that have a subset of these are hyperprogression disease, but these are patients who are not responding to immunotherapy. They're developing poor overall survival, poor progression-free survival. The antithesis is you see that blue line at the top. I think it's blue. These look like stage 1 patients. So we identified really two unique subsets, patients that are absolutely responding, a couple intermediate groups, and then a subset of group of patients that are not responding to therapy. And I'm only showing the OS data, PFS, those are the same thing. And this model validates, this algorithm validates in a test cohort and as well as an external VA cohort. And what's really, and we also applied this to a variety of different other, just basically available data sets that we had to see, is this a pan model? And over, we see that our radiomic feature reproduces, that it's not only predictive, but also prognostic. So, and then finally, we actually looked at the biology. Can we map this radiomic feature that we identified? Do we know what the biological underpinning of this? And so we had a radiogenomic data set of radiomic features and microarray data, and we found that it's associated with CA9, which is very provocative because we know that CA9 is upregulated in hypoxic tumors. And a hypoxic tumor are most likely not to respond to therapy. This is the case for TKIs, cytotoxic therapy, as well as immunotherapy. So we have a very interesting findings of a potential pan-radiomic feature. We call it an avatar because in our radiomic pipeline, we get rid of redundant and non-reproducible and correlated features. This one single feature represents an avatar of about 12 different features. So it's really getting down to a very parsimonious, simple model algorithm that would predict outcomes. So I'm right on time. I'm sorry. I try to catch up. I really want to thank a variety of people from all over the country, all over the world that are involved in this research. Bob Gillies, who couldn't make it today, I'm presenting on behalf of our team. We've been working together for about 10 years in this field. A lot of colleagues from China and our computer scientists at USF. Thanks.